Balls of Fire, The Science of Life and Death, Chapter 18, The Diamond Hearts. From a 21st century view, selecting this Diamond Heart fantasy draft team considers Time's legacy of scientific thought as well as the Isis thesis, which shows the ancient Egyptians understood our contemporary sciences. The views here support quantum theory and add credence to the Isis thesis in the face of its opposition, as generally represented by the Thunderhead team. The rising scientific awareness today is that the quantum world, ruled by quantum mechanics, with its equations resembling the kinetic molecular theory, orders our classical cosmos, ruled by general relativity, according to Einstein's theory of gravity. Other counterintuitive science includes physicist John Wheeler's delayed choice experiment, quantum non-locality, or being everywhere, and entanglement, the laws of Newton, Einstein, Maxwell, and Schrödinger permitting time reverse, and the mathematical equations of Einstein and current science that support the expanding/collapsing potential of our cosmos. Also relative are the findings of the International Human Genome Sequencing Consortium and the International Microbiome Sequencing Consortium. These are only a few of the modern scientific discoveries and experiments discussed in the 12 articles that support the following semiotic argument of the Isis thesis based on a unified interpretation of 870 ancient Egyptian signs. In brief, the Egyptian afterlife happens in the quantum mesocosm of microbes. Ancient Egyptian deities represent viral and bacterial genes, bits of DNA, and proteins. This knowledge, carved on the walls of the pharaoh's pyramids and tombs, and the bottoms of the nobility's wooden coffins, represents human deities functioning as viral and bacterial genes and proteins along the ancient glycolysis fermentation gene expression network present in our cells. The ancient network is also present in the common E. coli bacterium that houses its viral companion phage lambda that dominates the E. coli bacterium with its genetic switch between two lifestyles. When lambda chooses lysogeny, it invades the cell and falls asleep on the bacterial DNA, replicating silently with the bacterium. When lambda chooses lysis, it switches to an active attack on the bacterium, taking over the bacterium's replication machinery to clone viral particles. Along with a concise description of this powerful genetic switch, Egyptian texts support that bacteriophage lambda is the quantum world heart of our cosmos and the last universal common ancestor of human beings. Further, the ancient Egyptian texts explain that human consciousness and existence result from the viral lifestyle of lysogeny. Then, at a death transition, a human has a quantum choice for continuity of mind or DNA transformation to a quasi-hybrid being that is a higher ordered, crystallized state of existence created by the viral lifestyle of lysis. Because the laws of our cosmos with its holographic mode of operation are ordered by the laws of the quantum world, a human agent with knowledge can collapse the cosmos to its early state, as supported by Wheeler's observer participancy experiment and human potential for quantum state reductions. This amounts to the human observer hitting the switch for viral lysis. This brief summary of the Isis thesis, the selected players who have theories, experiments, and observations supportive of the thesis are lined up in the following batting order. First base, we have theoretical physicist John Wheeler, 1911 to 2008. Recent research is supporting our impressive potential for quantum choices. For example, physicist John Wheeler explains our potential for observer participancy based on his delayed choice experiment. In broader terms, we find that nature at the quantum level is not a machine that goes its inexorable way. Instead, what answer we get depends on the question we put, the experiment we arrange, the registering device we choose. We are inescapably involved in bringing about that which appears to be happening. Mind is central to the complete cosmic system because the act of observation permeates quantum mechanics. according to physicist John Wheeler's observer participancy principle. Because of quantum entanglement, which entails the phenomenon of non-locality, objects can become linked and instantaneously influence one another regardless of distance. This is because they are non-local or everywhere at once in the quantum domain. Physicist John Wheeler explains our potential for observer participancy, suggesting we must abandon causality or cause to effect. In a delayed choice experiment, one discovers that a choice made in the present has past consequences in the earliest days of our cosmos when life on Earth did not exist. As confirmed by Wheeler's delayed choice experiment, we can bring about that which appears to be happening. This is because the past exists only in the present. Wheeler explains we have to move the imposing structure of science over onto the foundation of elementary acts of observer participancy. This means that quantum mechanics lionizes the role of the individual observer. Think about that. And so the unaware individual human being possesses an immense power of choice in both our classical world of matter and the quantum domain of proteins and genes. As Wheeler explains, the world of time and space is a system of existences that has given birth to observer participants who act in a network of elementary quantum phenomena. The universe is not a machine. The quantum is the foundation of physics, and it operates with a pure yes-no character. Time is derived from profound considerations of a quantum flavor, and the physical continuum is myth. The ancient Egyptian, early Chinese, American Navajo, and Aztec texts, the act of observer participancy results in an act of cosmogenesis because the world represents the quantum networking of two viral proteins in their energy landscapes. Because our macrocosmic world is quantum mechanical, understanding Wheeler's observer participancy principle is the first step to winning the game of the centuries. Second base, biologist Lynn Margulis, 1938 to 2011. Lynn Margulis is a good pick for second base because she understands the fundamental impact of the microbial mesocosm on human evolution, life, and death. In Microcosmos, 1986, Margulis and Sagan explain how hustle bustle bacteria invented life's chemical systems, leading to fermentation, photosynthesis, and oxygen breathing. After we die, explain Margulis and Sagan, we return to the microcosm. This is the same message of ancient Egyptian as well as early Chinese and Navajo texts, but these cultures offer us environmental survival tips and other signs for the evolutionary process using the ancient glycolysis fermentation pathway in our cells. Before the advent of oxygen, the ancient glycolysis fermentation pathway is believed to be the source of energy production for earlier organisms such as bacteria. Ancient Egyptian texts support this scientific belief. Now, the unexpected coincidence is that this ancient pathway in the gram-negative E. coli cell has a gateway called the lamporin, the outer membrane protein producing open water-filled pores, that is, the selective attachment site for phage lambda. Glucose is the preferred carbon source for the cell. However, when glucose is limited and the cell is starving, the lamporin will open its gates across the outer membrane to other polar nutrient solutes. So our cells have the metabolism adaptation process for the possible emergence of higher levels of organization involving sugars other than glucose, such as maltose and lactose. Because of the metabolic regulation mechanisms in our cells, we are well organized for survival. Margulis and Sagan explained the ancient glycolysis fermentation pathway in our cells, saying, quote, fermentation was never lost. When we exert ourselves, for example, by running up the stairs, our cells momentarily bypass their typical oxygen-mediated metabolism and revert to the ancient fermenting mode. Although less efficient means of deriving ATP, this fermentation metabolism is still with us. Bacteria recycle the substances in our bodies, and Margulis and Sagan understand that the microcosm is evolving as us. However, we are, quote, locked into our species because we trade genes vertically rather than horizontally like bacteria. The result is that while genetically fluid bacteria are functionally immortal in eukaryotes, sex becomes linked with death. As Margulis and Sagan support, all organisms evolve through symbiosis, the coming together that leads to mutual benefit through the permanent sharing of cells and bodies. Symbiosis also explains the exchange of DNA between two different species that produces the hybrid. Margulis' ideas on symbiosis and microbiology have contributed to evolutionary theory. She has broken down biological barriers in a manner similar to second baseman Jackie Robinson, who broke through racial barriers with a career .983 fielding percentage at second base. Shortstop, Guenther Witzeny, philosopher of science. Austrian philosopher Guenther Witzeny promotes a theory of communicative nature, showing that interactions within cells, between cells, and within living individuals function by sign-mediated interactions. His transdisciplinary research and approach includes philosophy, biology, and semiotics. He recently applied his ideas on biocommunication to current research about viruses and bacteria at a Cold Spring Harbor laboratory meeting on telomeres and telomerases. Guenther supports that our non-coding repetitive DNA, what has been referred to as DNA or junk DNA, comprises non-coding regions necessary for higher order regulatory and constitutional functions related to protein structure. These repetitive genomic elements, introns, are viral in origin. Telomerase is a reverse transcriptase, a key feature of these repetitive elements, while telomeres are repeated elements protecting our eukaryotic DNA chromosome ends from genetic parasites. Recall that technocrat Aubrey de Grey would like to remove telomerase from the human genome. At this presentation, Guenther used a biosemiotic approach, that is, biological communication processes as sign-mediated interactions. He explains how biosemiotic perception of telomeres is a view of viral genome organization that considers the idea of mutations as the natural genome editing competences of viruses. So a mutation is reconsidered as functional innovation or viral genome inventiveness. Guenther shows how a dormant, persistent virus within the host is non-destructive, allowing the virus to transmit complex traits to the host cell. This improves the host cell's evolutionary potential that could lead to a new species. Some of this novelty includes gene functions such as immunity, gene silencing, and recognition functions such as receptors. Guenther's list of viral inventions is impressive and surprising. RNA and DNA replicase, polymerase, integrase, methylation, histone modification, the innate immune system, the adaptive immune system, larvae, egg, placenta, flowering plants, skin, dermal glands for mucus, milk, linear chromosomes, natural DNA repair techniques, bilayer nuclear envelopes, nuclear pores, division of transcription and translation, tubulin-based chromosome duplication, cartilage bones, and viviparous animals. Guenther also informs us that every key feature of our eukaryotic cell nucleus is present in a DNA virus, such as the prokaryotic phages, cyanophages, Q-bacteriophages, and archaeophages. This supports the ISIS thesis interpretation regarding the evolutionary precedence of eubacteriophage lambda with its double-stranded DNA. This suggests that a double-stranded DNA molecule like phage lambda is the progenitor of our eukaryotic nucleus. Reconstructing early events in the transition from prokaryotic to eukaryotic cells is problematic for evolutionary biologists. Some believe eukaryotes were born out of a stepwise symbiotic union of prokaryotic cells, while others believe that the ability to phagocytose, eat prey, led to the origin of eukaryotic features prior to endosymbiosis. Perhaps the transition from prokaryotic to eukaryotic cells was the result of a lambda prophage in a prokaryotic cell evolving into the eukaryotic nucleus. Let's take a closer look at Witzeny's research on telomeres and telomerase, key features found in viral genomes. Whitney believes that they are a natural genetic engineering tool with context-dependent functions. He explains that research shows life must be viewed from the crucial role played by viruses. In other words, a virus is a living being and must be placed at the beginning of all life long before cellular life evolved. A virus integrated into the hosting organism is beneficial, for the process broadens the host's evolutive potential and may promote the formation of new species. Non-coding introns are viruses. Whitney concludes, in eukaryotes, telomerases and other reverse transcriptases act as endogenous viral competences. So our eukaryotic viral genome, with its telomerase and reverse transcriptase, is a potential toolbox for a new species. Gunther also is a good pick for shortstop due to his following statement regarding self-reanimation of virus. By mixing defective viral genomes, it is possible to recombine a full function virus again. Their highly conserved repair and recombination capacity enables them as the only one living beings which can reanimate as living being after their death. This comment about defective viral genomes points a finger at our non-coding 98% junk DNA, a cemetery of viral genomes. So a symbiotic union of DNA with a virus may not be a bad idea at a human death transition, for it may fashion what ancient Egypt suggests, a full function virus. Add to this that Gunther discusses the amazing functionality of telomeres as well as telomerase, a natural genetic engineering tool that the machine dream pitching team would like to delete from our genome to cure cancer. However, these natural genetic genome editing competences are the original skills of viruses and may be the exact toolkit necessary for horizontal gene transfer and viral transformation at a human death transition. One must reconsider the value of the large repository of viral DNA in our genome and microbiome. Working with Louis P. Villarreal, the director of the Center of Virus Research at the University of California at Irvine, Gunther argues that viruses are a major creative force in the evolution of life. Louis had presented a paper at the American Philosophical Society entitled, "Can Viruses Make Us Human?" He listed the following traits provided to eukaryotes are cell type by viral genes in the evolution of complexity. Eukaryotic nucleus and flowering plants, the adaptive immune system in animals, live birth, viviparous placental mammals, multi-membrane bound separation of transcription from replication, chromatin proteins, linear chromosomes with telomere ends, DNA-dependent RNA polymerase, enzymes that modify mRNA in a eukaryotic specific way, complex role of tubulin in the separation of eukaryotic daughter chromosomes, and the dissolution and reformation of the nuclear membrane. Louis Villarreal and Gunther Whitney are witnesses to the value of the essential traits we have received from viruses. Although lytic viruses are associated with disease and death, viral bacteriophages ground and nourish the matrix of life with their symbiotic ability for horizontal gene transfer. When some traditional researchers popped up 4 arguments why viruses should be excluded from the category of life: their inability to self-replicate, their polyphyly, their cell origin of their genes, and their genetic vitality. In an article, Lewis and Gunther cut off all 4 arguments by presenting current knowledge on the key roles of viral interactions. They explain that the properties of our eukaryotic nucleus are attuned to derivation from a stable, persistent DNA virus with linear chromosomes. Phage lambda is a ubiquitous virus with linear chromosomes. Luis and Gunther also discuss Patrick Fortere, who supports that RNA polymerase, DNA polymerase, and DNA helicase, agents that transcribe and replicate DNA in modern mitochondria, may also have viral origins. Viruses play an important role in the evolution of diversity as a driving force of all cellular life forms and essential processes. Also, increasing eukaryotic complexity correlates with expansion of non-coding DNA. So the non-coding DNA must be a useful toolbox for future morphogenesis. Also, the tailed icosahedral phages like phage lambda represent a huge family of viruses that can infect both bacteria and archaea, showing their incredible ubiquity. Louise poses the question as to why the evolution of higher genetic complexity is connected to non-coding virus-derived DNA that was formerly called junk DNA. The ISIS thesis suggests that this viral DNA is the necessary precondition or toolbox for human transformation by means of a bacteriophage at a depth transition, using the endogenous viral elements in the human genome. Also, the adaptive phage genome or phagome in our mammalian gut ecosystem is a potential genetic reservoir with functional genes that operate under stress-related conditions. Finally, viruses can create a new sequence de novo from the beginning, anew, supporting an emergence that may be similar to ancient Egypt's crystallized quasi-hybrid being, or perhaps Frank's time crystal. In the beginning, perhaps a bacteriophage entered a cell, became a prophage, and then developed into our eukaryotic nucleus. Third base, astrophysicist Jean-Pierre Luminet. According to astrophysicist Jean-Pierre Luminet of the Paris Observatory in France, the cosmic microwave background, CMB, or radiation left over from the Big Bang, points to our cosmos having a complicated dodecahedral shape within a hypersphere. This topology has also been described by the early 20th century mathematician Henri Poincaré, as well as Plato millennia earlier. If Luminet's findings turn out to be true, the dodecahedral shape that is dual to the icosahedron suggests that the microstructure that left this signature at the Big Bang origin of time may be phage lambda, for the virus has an icosahedral head enclosed in a sphere. The basic icosahedral phage conformation consists of 20 equilateral triangles arranged around the face of a sphere. The icosahedron is a triangular model of fivefold symmetry in 3 dimensions, having 20 faces, 12 vertices, and 30 edges, while its dual, the dodecahedron, has a pentagonal shape with 12 faces, 20 vertices, and 30 edges. Let's reflect on Luminet's ideas in the wraparound universe. A hall of mirrors produces an optical illusion, a multiplication of images. Astrophysicist Jean-Pierre Luminet observes that mirrors not only reflect images, but they also carry the secrets of the infinite. For example, a room with mirrors on all 4 walls and the ceiling allows us to see multiple reflections infinitely far in every direction. Based on his research, Luminet explains that cosmic space may be a similar illusion to a hall of mirrors. This universe of ghost images will be created by light trajectories following the folds of a wraparound universe. Luminet believes that global cosmic space is wrapped and finite with negative curvature. Luminet explains that a dozen gravitational celestial mirages have been detected. He claims topological mirages created by the topology or shape of a wraparound universe may produce image multiplications, which are much more surprising than even the gravitational mirages. Interested in the shape or topology of space, Luminet suggests that visualizing microscopic space as an ocean enables one to understand the structure of space at small scales relative to unifying fundamental interactions according to quantum gravity theories. This illusion is similar to a hall of mirrors. However, it also sounds as if we could be living in a cosmic crystal or a crystalline protein folding funnel. In an article entitled Science, Art, and Geometrical Imagination, Luminet informs us that the symmetries of the dodecahedron are found in science and art. For example, Plato considers the dodecahedron, the shape closest to a sphere, as the solid representing our universe. Luca Pacioli, a Tuscany mathematician and artist, as well as collaborator with Leonardo da Vinci, was also impressed with the polyhedral shape and the golden ratio. It is also interesting that the long spiral of the Egyptian Eye of Horus is similar to the Fibonacci spiral that converges on the golden ratio, phi, which relates to the icosahedron, pentagon, DNA, and Kerr black holes. As Dr. Mario Livio, senior astronomer at Hubble Space Telescope Science, explains, spinning black holes, called Kerr black holes after the New Zealander physicist Roy Kerr, can exist in two states: one in which they can heat up when they lose energy, negative specific heat, and one in which they cool down. They also can undergo a phase transition, similar to the freezing of water from one state to the other. The transition can take place only when the black hole reaches a state in which the square of its mass is precisely equal to phi times the square of its equal angular momentum in the appropriate units. Now, the eye of Horus numbers half, 1/4, 1/8, 1/16, 1/32, and 1/64 are equivalent to black hole resolutions, determining the position of the inner event horizon via the apparent horizon. An apparent horizon is a structure of space-like hypersurface that can be determined locally in time. However, event horizons are global structures that cannot be detected during a time evolution. The eye of Horus numbers almost converge on zero, or the minimum radius of the inner horizon, necessary to avoid the repulsive ring singularity, which has a non-zero radius. As mentioned, the icosahedron and dodecahedron are dual polyhedra because the centers of the faces of a regular dodecahedron are the vertices of a regular icosahedron, and the center of the faces of a regular icosahedron are the vertices of a dodecahedron. With his ability to see the universe as a crystal, Luminet is going to hit more than one home run in the game of the centuries and astrophysics. He is as good as Brooks Robinson, who received 16 consecutive Gold Gloves. Robinson played for the Baltimore Orioles from 1955 to 1977, and he was a great fielding third baseman. With his wraparound skills, we expect Luminet to be cosmic at third base. All right, so where we at? All right, so now we went from first base to second base to third base. Let's get to fourth base. So fourth base is going to be talking about where we at? Yeah, so fourth base is going to be talking about theoretical biologist Stuart Kauffman. So Stuart Kauffman is an influential force in the field of biology. Like Tiger right fielder L. Kaline, who hit his .399 career home runs, Kauffman has probably watched the same amount of proteins fold to their home plate native state. Kauffman deserves a wave place in a baseball biology hall of fame because he explores the forces for order that lie at the edge of chaos. Order is free in light of the science of complexity that involves the scientific trinity of Darwin's theory of evolution by natural selection, self-organization, and chance. Kauffman informs us that order arises from two different forms. The first is a low-energy equilibrium system, such as a ball in a bowl rolling to the bottom due to gravity, wobbling and stopping at equilibrium where no more energy is necessary. Similar to the ball in a bowl, Kauffman classifies viruses as low-energy equilibrium systems. Thus, viruses are complex molecular systems of DNA or RNA molecular strands that form a core around which a variety of proteins assemble to form tail fibers, head structures, and other features. In an appropriate aqueous environment, the viral particle will self-assemble from its molecular DNA or RNA and protein constituents, seeking a state of lowest energy, like the ball in the bowl. Because no more energy is necessary to maintain the virus, order is free, like it is in Frank's time crystal, which does not need an outside force of energy to keep it moving at minimum energy. Now, viral DNA self-assembly with organic human DNA is clearly described in ancient Egyptian texts as the merger of the sun god with the deceased person for an existence of millions of years in the early universe. This results in a crystal or a product similar to Frank's time crystal, Frank's time crystal, where order is free and life is not robbery. Kaufman's second form of order is a non-equilibrium system that requires a constant source of energy to sustain the order structure, such as a whirlpool in a bathtub or the great red spot vortex of Jupiter. This large vortex or storm system, existing for several centuries, is sustained by the dissipation of matter and energy. Nobel laureate Illa Prigogine used the term dissipative structure to describe such a non-equilibrium ordered system as the red spot, where the flux of matter and energy throughout the system generates its order. All known free-living systems, bacteria, insects, animals, humans, are composed of cells that are non-equilibrium dissipative structures. Even our biosphere is a non-equilibrium system using solar radiation. To these free-living systems, equilibrium means death. However, a virus is not a free-living, non-equilibrium system because as a parasite, it requires a host cell to reproduce. What is needed, explains Kaufman, is the establishment of general laws predicting the behavior of non-equilibrium systems. Kaufman believes that there can be laws of life or biology. Vast order arises naturally, and life exists at the edge of chaos, near a kind of phase transition between order and chaos. For instance, Kaufman considers the Pleuromonas, a simplified bacterium with a cell membrane, genes, RNA, proteins synthesizing machinery, proteins as living. He emphasizes that the vastly simpler virus is not alive, yet he queries the reader, why can't a system simpler than Pleuromonas be alive? Life is an expected emergent property of matter and energy, claims Kaufman, so we have a home in the cosmos. Perhaps Kaufman is aware of Frank's mathematical solution for a time crystal. Kaufman also proposes a potential universal law. It is far too early to assess the working hypothesis that complex adaptive systems evolve to the edge of chaos. Should it prove to be true, it will be beautiful, but it will be equally wonderful if it proves true that the complete adaptive systems evolve to a position somewhere in the ordered regime near the edge of chaos. Perhaps such a location on the axis, ordered and stable, but still flexible, will emerge as a kind of universal feature of complex adaptive systems in biology and beyond. According to ancient Egyptian, early Chinese, and Navajo texts, the axial rule is essential for the emergence of the human adaptive system at the polar axis of a cell, where the ordered regime is near the edge of chaos. Along with this potential universal feature related to the axis, Kaufman states that the genomic network is another example of order for free. Relative to the ISIS thesis, order arises naturally because it is guided by the viral genome activating the genetic switch to lysis within the ancient glycolysis fermentation pathway in a cell. Viruses, humanity, and Earth are co-evolutionary systems. The Earth is a non-equilibrium system invaded by solar radiation. On the quantum level, the Earth's cell is similar to a living whirlpool, a non-equilibrium bacterial system invaded by solar viral particles. Yet a virus is an equilibrium system using the non-equilibrium Earth cell to reproduce. Further, horizontal gene transfer mediated by a virus is what saves organic DNA from dissolution at death. At death, we need new traits to be fit or at home in the universe, so a symbiotic union, a horizontal gene transfer with a crystalline bacteriophage that looks like a ball of light, may provide exactly those elements our DNA needs to adapt to the quantum environment, especially if that virus is our ancestor, a double-stranded eubacterial virus like phage lambda with every feature of a eukaryotic cell nucleus. Kaufman states that fitness increases if a population uses mutation, natural selection, and recombination to climb to the peak on a well-correlated landscape. Recombination is useless on a random landscape. Also, the space of possible molecules is immense. A stable, low-energy equilibrium state is present in a assembled virus, the double helix of DNA or RNA, and in the folded protein encoded by genes. These stable structures are robust. Likewise, the ancient Egyptian system is not a random fitness landscape, for they describe mutation, recombination, and have left explicit instructions relative to lambda's genetic switch using game theory. We have a coupled self-organized system with CRO and C1 proteins competing and partnering for genomic binding to switch on their respective genes. This randomness is critical to the evolutionary assembly of complex organisms and the emergence of new organisms, for it forms a circle cycling back to the genome or home plate. Lambda, CRO, and C1 have correlated landscapes due to adjacent genes on the lambda chromosome, but the proteins fold differently, and CRO is an initially disordered protein like our non-coding DNA. Amidst this chaotic genomic network with its functional genetic switch is the ordered regime of CRO protein's native state. Kaufman states the deepest source of such landscapes, non-random, may be the kind of principles of self-organization we seek. Here is one part of the marriage of self-organization and selection. He believes that we need a new conceptual framework that will allow us to understand the evolutionary process in which self-organization, selection, and historical accident play roles. He suggests that evolution can assemble a maximally compressed program or organism by first evolving a redundant program or organism and then squeezing it down to maximal compression. Certainly, our genome is loaded with redundant DNA that may satisfy these requirements, especially the abundant reverse transcriptase and telomerase with its reverse transcriptase. Like Al Kaline, who won 10 gold gloves and 15 all-star team selections, Tiger Kaufman has a model of consistency for excelling in biology as well as the sporting dexterity to perform in right field for the Diamond Hearts in the game of the centuries. Catcher, virologist Luc Montagnier, left field wave geneticist Peter Garriev. For a long time now, Luc Montagnier, 1932 to 2021, has been catching biophysical fastballs based on experiment and theory that often result in controversial discoveries. Both virologist Luc Montagnier and wave geneticist Peter Garriev are involved in challenging scientific pursuits that may provide new strategies for viral diseases such as cancer and HIV. Peter Garriev is director of the Wave Genetics Institute, Russia, Moscow. The French virologist Luc-Antoine Montagnier is a joint recipient with François Barrère-Senosse and Harad Zor-Hassan of the 2008 Nobel Prize in Physiology or Medicine for his discovery of HIV. In 2010, Luc became a full-time professor at Shanghai Jiaotong University in China, where he explores electromagnetic waves emanating from the DNA of pathogens. To understand Luc's focus, let us first consider the nature of light, or photons, that travel in the form of a wave. Photons, or electromagnetic radiation, are a form of radiant energy moving through space via electromagnetic waves and or particles. So we have the simplest formula of light equals photons equals electromagnetic radiation. Now, based on earlier Russian research, Peter Garriev and his colleagues have shown that genes or DNA have electromagnetic wave representation. Then, in 2009, Luc Montagnier and his colleagues shook up the theoretical underpinnings of chemistry by suggesting that DNA sends electromagnetic imprints of itself into distant cells and fluids. Further, these researchers detected electromagnetic signals from bacterial DNA, such as E. coli, in water. The researchers believe that this is a novel property of DNA. Some sequences can emit electromagnetic waves in resonance after excitation by the ambient electromagnetic background. This ambient background is the Earth's 7 Hz naturally occurring oscillation of its electromagnetic waves. Human brain waves also oscillate in the same range. How does this research support the ISIS thesis? Ariev and Luke's experimental research supports that genetic information can function not only on a material level, but can also function in a wave field or in an electromagnetic field. Similar to Geriev and Luke's views, the ancient Egyptian texts indicate that our world is holographic and ordered by quantum electromagnetic fields that represent microbial DNA. To explain, the ancient texts demonstrate that the Earth's electromagnetic field in watery space is a sign of the genetic code of E. coli bacterium on the quantum level. The researchers finding that bacterial E. coli DNA can send electromagnetic imprints of itself into distant cells and fluids corroborates the ancient Egyptian knowledge, considering the holographic principle. If the Earth's magnetic field represents E. coli DNA on the quantum level, then the Earth's magnetic field becomes the guiding platform for traversing the E. coli chromosome, as the ancient texts explain. What stimulates or emits the bacterial electromagnetic waves is the Earth's 7 hertz electromagnetic waves, and human brain waves oscillate within the same range. So the ISIS thesis model of the Earth as an E. coli cell is consistent with this research. As some scientists have commented, the stars may represent DNA written across the crystalline cosmos. Now, Geriev believes that our genome represents two intimately linked substances. The 98% junk DNA is a supercode that codes at a higher wave level than our 2% material coding DNA. Although the two substances are intimately linked, the EI level is the leading level grounded in the 98% of our DNA with viral elements. In the ISIS thesis interpretation of our DNA text, the interplanetary magnetic field of the sun is the sign of the viral DNA of bacteriophage lambda. In comparison to Gariel's findings, this interplanetary magnetic field will represent Gariel's 1-to-1 level. The ancient Egyptian instructions for transformation explain how to, quote, cross over or recombine with viral DNA. Find the viral polar gateway into the cell, the lamporin of E. coli, and enter the ancient glycolysis fermentation pathway for transformation to a quasi-hybrid being. Put simply, Gariev's ideal 98% of our viral genome may be a super gene continuum that ensures the development, life, and programmable death of living things. However, the ideal 98% also conveys a survival message of viral horizontal gene transfer and transformation. This suggests that human consciousness with a memory of the genetic survival message is the switch hitter that activates the lactic genetic switch for viral replication. One could say that the ancient Egyptian dead king functions in a fashion similar to the phantom DNA effect. Granted, these claims are slippery fastballs, but let's take another step deeper into Gariev's research. So we'll stop there. I will pick up with the rest of this in another video. So I'll see you in the next one. Peace.